Good morning. Today we'll discuss an interesting topic, blood type or blood group. What is blood type or blood group? A blood type or a blood group is a classification of blood based on presence or absence of antibodies and inherited antigenic substances which are present on the surface of red blood cells. Observe the first column. If your blood group is A, your red blood cell is surrounded by antigen A. If your blood group is B, your red blood cell is surrounded by antigen B. And blood group is AB, both the antigens, antigen A and antigen B are present. If your blood group is O, there are no antigens. So now the question arises, what are antigens? Antigens may be protein, carbohydrate, glycoprotein, or glycolipid depending upon the blood group system. Observe the second row. If blood group is A, antigen is A, antibody is B. And if blood group is B, antigen is B, antibody against B antigen is anti-A. Observe the AB blood group. There are no antibodies. If there are no antibodies, AB can receive blood from A blood group, B blood group, or O blood group. That's why AB blood group is considered as an universal recipient. Observe the last column, that is blood group O. Both antibodies are present, antibody B and antibody A. But antigens are absent. Therefore, group O can donate blood to ABO blood group. That's why group O is considered as universal donor. So blood group is classified, it falls into two groups. Observe the last line, ABO blood group, and one more is RH blood group. Now we'll understand the concept of blood group phenotype as well as genotype. Alleles. What are alleles? Alleles refers to one of two or more genes which occur alternatively at a given locus on a chromosome. It may occur in pairs or maybe multiple alleles. For example, if you observe our blood group, that is ABO blood group, the ABO blood group is controlled by the gene I. Just observe the red blood cell. It is surrounded by a plasma membrane, which has sugar polymers that protrude from its surface. The kind of sugar is controlled by gene I. Both IA, IB produces a slight different form of sugar, while allele small I does not produce any sugar. If you observe the first line, IA and IB are dominant alleles and small I is recessive allele. Observe the table now. If IA is from parent 1, IA is from parent 2, then the genotype is also A and blood group is A. If IA and IB are present, blood group is AB. Observe the third row. One is dominant IA and other one is recessive small I. But dominant character is expressed, therefore blood group is A. If IB and IA are present, both are dominant, the blood group is AB. IB, then again the option is B blood group. And observe the last but one column. The blood group is, allele is IB and small I. B is dominant over I, therefore the blood group is B. Last column, there are no dominant alleles, only recessive, therefore the blood group is O. In this way, ABO blood group is controlled by the allele I. Next, observe ABO inheritance pattern. So we already discussed if the blood group from parent 1 and parent 2 are A, then child's phenotype is also A. And if parent 1 is A, parent 2 is O, then the possible outcome is A. But observe the third and fourth, both are dominant. A is also dominant, B is also dominant. So both the characters are expressed. The child's phenotype is AB. This phenomena is called codominance. A is dominant, B is dominant, both are expressed. Therefore, the phenomena is called codominance. Both the traits are expressed. Gene A is inherited from one parent, gene B is inherited from other parent. The child's phenotype and the blood group is AB blood group. And remaining, if B and B, then the option is B blood group. B and O, then also B blood group. And if there are recessive alleles, that is O, then the child's phenotype belongs to O blood group. One more interesting factor is the presence of RH factor, RH inheritance. I mean, A positive blood group, B positive blood group, or A negative, B negative, AB positive, AB negative, O positive, O negative. This positive and negative are assessed based on the presence of RH factor. RH factor is inherited from our parents, but it is totally independent of ABO blood type alleles. It depends on the dominant rhesus D antigen. Just observe the first table. If both parents are Rh positive, then child's phenotype is also Rh positive. If one of the parent is Rh negative and Rh positive is the dominant gene, therefore the child's phenotype is also Rh positive. And in one more condition, parent 1 Rh negative, parent 2 Rh negative, so child's phenotype is also Rh negative. And if you observe the genotype, possible genotype for Rh factor, that is the second table, if our blood group is Rh positive, if our blood group is positive, then the genotype may be Rh positive, Rh positive, or Rh positive and Rh negative. Rh positive is the dominant rhesus D antigen, therefore only positive is expressed. If our blood group is negative, then the possible genotype is Rh negative and Rh negative inherited from parents. If both are recessive, then the child's phenotype is also Rh negative. So depending on the rhesus factor, blood groups are classified into positive and negative. Now we'll discuss one of the rarest blood group, Bombay blood group. First question comes to your mind, why it is called Bombay blood group? This blood phenotype was first discovered in Bombay by Dr. Y.M. Bhinde in 1952. Since it was first discovered in Bombay, the blood group is termed as Bombay blood group. What is Bombay blood group? Just observe the purple box. If there are no antigens on RBC, particularly if there is no H antigen on RBC, then it is considered as Bombay blood group. What is H antigen? H antigen is a precursor molecule. It is required for the synthesis of blood sugar. That is, if it is combined with Ia, then the blood group is A. With IB, blood group is B. If there are only H antigens, both are recessive and the blood group is O. But if there are no H antigen, if RBC is lacking H antigen, then it is called Bombay blood group. Observe one more small green box. It is the rarest blood group. Only 4 per million of world population may have Bombay blood group. And now question arises whether Bombay blood group people can donate blood to other blood groups. Yes, Bombay blood group people can donate blood to ABO blood group. Why? Because there are no antigens. But one interesting factor is Bombay blood group person cannot receive blood from any other blood group. Why? Because Bombay blood group persons the blood contains antibodies against A, antibody against B, antibody against H. Therefore, they cannot receive blood from ABO blood group. So what is the genotype of Bombay blood group? The genotype is small hh. Both are recessive, homozygous recessive, rarest condition, rarest possible case. Now observe the green box crossing between male parent and female parent. Both are carrier of recessive allele small h. And observe the chances, less possible chances. If there is homozygous recessive HH, then only the blood group is considered as Bombay blood group.